Today's virtual lecture will be on the one and only topic, the nucleolus. We have 4 students that will be answering what is the nucleolus, why it is important, what molecules are generated in the nucleolus, what enzymes are used inside the nucleolus to make other enzymes, and what happens to ribosomal RNA once synthesized.
Hi, my name is Vienna Martinez, and I'll be explaining what the nucleolus is and why the nucleolus is important. So, what is the nucleolus? The nucleolus is a large rounded structure that is located inside the nucleus. It can either be found in a plant cell or an animal cell, but not in a prokaryotic cell. In the nucleolus, there are clusters of chromosomes which contain genes for ribosomal RNA. The nucleolus is very important and essential in its cell duties, so we'll be discussing that right now. Why is the nucleolus important? The duties that the nucleolus has is that it makes the rRNA, which then leads to the synthesis of ribosomes and then leads to the synthesis of proteins. In this structure, ribosomal rRNA is transcribed and ribosomal subunits are assembled. Once these ribosomal subunits are made, they are delivered to the rest of the cell where they make complete ribosomes. And these ribosomes are then used to make proteins. So essentially, the nucleolus plays a vital role in protein synthesis as well.
So, to go off of what my previous colleagues said, the nucleolus is a large structure within the nucleus where ribosomal RNA is transcribed and where ribosomal subunits are assembled. The nucleolus can be found inside animals as well as plants. However, the nucleus cannot be found inside a bacterial cell. This is because the nucleolus can only be found inside eukaryotic cells, not prokaryotic. So, again, one of the most prominent structures within the nucleus is the nucleolus, which is the site of rRNA transcription and processing and of the ribosome assembly. As discussed inside our cell biology textbook, the cells require a large number of ribosomes to meet their need for protein synthesis. So, for example, inside our cells, eukaryotic cells, we can have about 5 to 10 million ribosomes that need to be synthesized each time the cell divides. So the nucleolus basically becomes a ribosome production factory and is designed to fulfill the needs for a large-scale production of the rRNAs and the assembly of the ribosomal subunits. So as a reminder as well, the nucleolus is not surrounded by a membrane, okay? Instead, it is organized around the chromosomal regions that contain genes for rRNA. So a great example that occurs inside the nucleolus is chromosome organization during interphase. The nucleolus is where parts of different chromosomes that carry genes encode the ribosomal RNAs and they can cluster together. So here they're synthesized and they combine with proteins to form ribosomes, which is the cell's protein synthesizing machines. Ribosomal RNAs play both a structural as well as catalytic roles within the ribosome in the nucleolus. So, as a recap, what molecules are found inside the nucleolus? The nucleolus is where ribosomal RNAs are synthesized. Which are then combined with proteins to form ribosomal subunits. Which then can be exported outside the cytoplasm, where the cell can then carry out its different functions with these ribosomes.
Hi, my name is Joshua Preet-Sadhu, and I'll be touching on the importance of why enzymes are used to make enzymes. So in the nucleolus, ribosomes are the enzymes used to make enzymes. Ribosomes are biological molecules made up of RNA and ribosomal proteins. Ribosomes are responsible for making the proteins that are essential for cells. The ribosomal RNA is transcribed from the nucleolus, which is where ribosomes are synthesized in a cell. Eukaryotic ribosomes are essential to two subunits that are made up of 4 distinct ribosomal RNAs. Basically, ribosomes are enzymes that are not made up of proteins. Even though only a few enzymes are made up of proteins, ribosomes are one of them because they're made up of RNA and not polypeptide chains. This is also why ribosomes are called ribozymes. So now we will be moving on and creating a chart comparing ribosomes within prokaryotic cells and eukaryotic cells. In prokaryotic cells, such as bacteria, the ribosomes tend to be small and the rRNA is organized into 3 strands. While in eukaryotic cells, such as humans, the ribosomes are much bigger and the rRNA is organized into 4 strands. Also, prokaryotic cells only tend to have 70S ribosomes. Meanwhile, eukaryotic cells have different types of ribosomes. Lastly, we'll be talking about prokaryotic cells having 3 types of rRNAs and 50 types of proteins. But eukaryotic cells only have 8 types of proteins and 5 types of rRNA. So this chart here explains prokaryotic cells and eukaryotic cells comparing the ribosomes, but let's not forget that the nucleolus does not exist in prokaryotic cells.
I'm Natalie Diaz, and I will be speaking about what happens to ribosomal RNA once it's been synthesized. rRNA associates with a set of proteins to form ribosomes. These complex structures, which physically move along an mRNA molecule, catalyze the assembly of amino acids into protein chains. They also bind tRNAs and various accessory molecules necessary for protein synthesis. Ribosomes are composed of a large and small subunit, each of which contains its own rRNA molecule or molecules.
So I will be recapping why the nucleolus is so important to us and our cells. What is the nucleolus? It is a large, rounded structure that is located inside the nucleus. And it is found in eukaryotic cells, but not prokaryotic cells. So therefore, it is only located in either plants or animals. The molecules in the nucleus are ribosomal RNAs, as well as proteins, and ribosomal subunits. Because the ribosomal RNA is combined with the proteins to make these ribosomal subunits. And then they can be exported into the cytoplasm, where the cell can carry out its different functions with these ribosomes. The ribosomes are actually the enzymes in the nucleolus. They act as catalysts to make proteins. So what happens after the molecules and enzymes are synthesized in the nucleolus? Well, the ribosomal subunits were the last things to be made in the nucleolus. These ribosomal subunits are exported into the cytoplasm to form ribosomes. The ribosomes are catalysts, and so they can catalyze the amino acids to a protein chain. And essentially, they help out with making proteins. So hopefully now everyone realizes why the nucleolus is so important and why it is an essential component to our cells.